Hello everybody and welcome back. So, in the last lecture I had given a problem, numerical problem, and today I will quickly show you the answer. It is very easy actually. The problem was that if you do a PCR, let us say you have a double stranded DNA for which you are doing the PCR, and after about 10 cycles, then you get a certain quantity of the DNA, and the measured optical density or the measured UV absorption at 260 nanometer was coming as approximately 2. That was the experimental quantity that you have obtained after the cycle. Now, the question is, if at the beginning, if you have the absorbance we call A1, and if this is now A2, then the question is what was A1? That was the problem, and I had given I think few data also, that the molecular weight or the molar mass of the DNA, given DNA, was around let us say 20 kilo Dalton, and the calculated epsilon value at 260 nanometer was approximately, this is the molar extinction coefficient, 4 into 10 to the power 5 liter mole inverse centimeter inverse. So, what is A1 actually? So, you must have already done this is very easy, and this data I guess you probably will not require them at all, they were meant to confuse you. So, if you want to calculate what was the basic equation? Basic equation is the Beers-Lambert's law, absorption equal to epsilon into C into L. This A is, as you know, A is the absorbance at a certain wavelength. In this case, this is 260 nanometer, because we are dealing with DNA, and DNA absorbs maximum at 260 nanometer, and this is unitless, there is no dimension for absorbance. Epsilon is the molar extinction coefficient, and it has the standard dimension of liter mole inverse centimeter inverse, and this is a characteristic of a molecule. So, you can calculate epsilon for depending upon the DNA sequence, but it is kind of a characteristics. So, it does not change over dilution, or it does not change over, if you change the concentration of your sample, epsilon remains intact. So, it is a pretty much constant quantity and kind of a characteristic of the sample. C is the concentration of your DNA in this case, and L is the path length. Path length means the length through which the light travels. What is the distance that the incoming light travels through your sample? So, ideally the path length is actually the length of your cuvette. If you are using, we of course have to use a cuvette to measure the UV. So, it is basically the dimension of the cuvette, and the most standard dimension of the cuvette that we use, or diameter of the cuvette that we use, is 1 centimeter. So, we have to measure basically the concentration first, or our target is to measure the absorbance at the beginning. So, if you start A1 equals epsilon, at the beginning let us say the concentration was C1, and L, you are using the same cuvette. So, essentially L is same for both cases. A2 is epsilon, does not change because it is the same DNA, let us say the concentration has been changed to C2 and L. Now, A1 by A2 basically comes down to C1 by C2. Now, so other parameters are not required in this case. So, what is A2? So, C1 and C2, is there any relation? So, C1 is the concentration at the beginning. What is C2? C2 has to be C1 into 2 to the power 10. This should be your C2, or 1 by 1024. You have to calculate your A1. Your A2 is given as 2. This is your A2, 1024. So, your calculated A would be around 1.9 into 10 to the power minus 3. So, A1 is 2 by 1024. I hope I have done the calculation correct, but this is it. From here you can get your A1. So, now let us move our attention to another important discovery, another important aspect, that is called the DNA sequencing. A highly important and highly fascinating subject is how to find out the sequence of a given DNA. So far we have seen the structure of a DNA. We know that it forms a double helix. We have seen what is the composition of the DNA, that it has 4 nucleic bases and it has a phosphate unit, it has a sugar unit. And we have also come to know that the DNA is the most important material in the cell, because it dictates most of the other cellular functions. For example, it dictates the synthesis of protein and it passes on the genetic information to the offspring. So, now the question is, what is the sequence of a given DNA? If you have isolated a DNA from a source, then what nucleobases are coming after one another? That is of utmost important, because if you consider a molecule, if I give you a molecule, maybe an organic molecule or an inorganic molecule, and I ask you to find out more about the molecule. So, the first thing you would like to know is what the molecule looks like, what is the structure of the molecule. So, once you find out the structure of the molecule, once you find out how the molecule looks like, what are the atoms involved, how the atoms are attached together, what are the functional groups that are present in the molecule, then only you can think of the property of the molecule. You can get some idea what this molecule is going to do, what can be the characteristics of the molecule. Same for DNA. So, once you understand the whole sequence of a gene, how the nucleobases are attached one by one, then you can figure out, or you can have some certain ideas about what should be the characteristics or what should be the property of the given DNA. So, and you remember I have talked about hair samples or the samples DNA isolated from the diseased cells that are of low quantity. So, that one main purpose of studying those genomes is of course to see their sequence. Once you know the sequence of a DNA, then of a diseased DNA, then you can understand whether there is any mutation or not, or even you can use the DNA to match one identity to measure or to find out the identity of the persons or identity of the species also. So, Human Genome Project was a very big project that actually has done this entirely. It was entirely about studying the whole sequences that are present in human genome. What are the variations that are present from one community to the other, and it was one of the biggest project that actually was carried out almost all over the world. So, it is of that importance. So, today we will see the methods through which you can sequence a DNA. Primarily, there are two most popular methods that are used for sequencing a DNA. One is the Sanger's method. Sanger's, this is called di-deoxy method. Second one is Maxam-Gilbert method. So, these two methods we will discuss. The first is Sanger's di-deoxy method. So, by the way, both of these methods have been developed much before the PCR technique was evolved or developed. For example, Sanger's method was developed in 1977. And both Sanger is Frederick Sanger actually. Frederick Sanger received the Nobel Prize. Sanger actually has received his second Nobel Prize along with Gilbert and I think Berg also in 1980, and again most importantly they received the Nobel Prize in chemistry. So, you can see the enormous contribution the chemistry has in biology or in understanding the biological molecules. So, we will start with the Sanger's method or Sanger's di-deoxy method. Sanger's method uses the same idea as DNA replication. Only difference is that it uses a single stranded DNA instead of a double stranded DNA. So, your target DNA is a single stranded DNA. So, this is your phosphate 5 prime end and this is your hydroxyl, this is the 3 prime end. Let us say this is your target DNA. And this is a single stranded DNA. So, it uses the single stranded DNA as the template, and most often you can use the prime strand or the leading strand 5 prime to 3 prime direction. It also uses a DNA primer. A primer unlike DNA replication in normal cell that uses the RNA primer, the Sanger's method uses a DNA primer. I am not writing the phosphate again and again. So, 5 prime, 3 prime and this is your DNA primer, which is complementary to a part of the target. So, that has to be 5 prime to 3 prime end, and it also uses the polymerase and a single polymerase, but it does not use Taq polymerase. They have used the normal polymerase, normal DNA polymerase called DNA polymerase 1. DNA polymerase 1. So, Taq polymerase was actually not discovered in Sanger's time. So, they have used the normal DNA polymerase, and since they are not using heat, since they are using only the single stranded DNA, the normal DNA polymerase works fine. And of course, you have to remember that all of these things actually happened before the PCR was developed. And plus if you add now the DNTPs, so Sanger has also used the all the 4 DNTPs. If you add all of this, what will happen? You will basically get the synthesis of the complementary strand. So, using the DNA primer as the template here, you can synthesize the whole of the complementary strand. So, if you use all these ingredients, that is what you are going to get. Now, it does not tell you the sequence of your DNA. So, nothing special here. What Sanger has done is that, so he has divided the sample into 4 different vials. So, if you have these, what is the composition now? You have your single stranded DNA, that is your target. You have your DNA primer, only one primer you had to use. You have the polymerase 1 and you have the 4 DNTPs. DATP, it is important actually, that is why I am writing DCTP, DGTP and DTTP. So, that is what you have in your mixture, and then Sanger has divided into 4 different vial. So, vial 1, vial 2, vial 3 and vial 4. So, vial. So, all of these vials essentially contain the same composition, the same thing, and if you allow it to carry on the replication, all of them will give you the same double stranded DNA. So, again nothing special here. Now, what Sanger has done, he has done something extra, and that is where it is a brilliant combination of organic chemistry and biology. So, along with all the 4 DNTPs, Sanger has used this molecule. This is nucleobase ATGC H O phosphate 5 prime, and I am not writing the whole of it, P and P triphosphate. So, in each vial he has added a bit of this molecule. This is called, you can see now, do you see the trick? See, this is, there is no hydroxyl group. So, it is a deoxyribose we have called when there was a DNA sugar and there was a hydroxyl group here. So, this is 2 prime, this is 3 prime. In this case, you do not have any hydroxyl group. Both of these hydroxyl groups have been replaced by just simply hydrogen, and that is why this is called di-deoxy. So, it is basically di-deoxy ribose sugar, this unit, and it is a triphosphate. So, just like DNTPs, it is a triphosphate. Now, why this molecule? What this will do? So, if you see, if you look at the reaction, by now I think you know it by heart, that when the polymerase was reacting, what was happening? You have the base B1 O, and here let us say the DNA primer. We have started with the primer, this is the last base of the primer with a 3 prime hydroxyl free. What was the reaction? That it was reacting with the DNTP. O phosphate, phosphate, phosphate. So, polymerase was catalyzing this reaction, this was eliminating out, and that is how you get the new phosphodiester bond O this, and then again the 3 prime hydroxyl of this B2 will be free and it will continue the synthesis. That was the normal reaction that happens or that is catalyzed by the enzyme polymerase during the replication process. Now, once you use the di-deoxy, what could happen? DNA primer, OH, and here instead of the DNTP, you have the DDNTP. So, I will forget otherwise, I am going back. Di-deoxyribose or ribonucleic acids is known as, is called abbreviated as DDNTP. So, DNTP is the deoxyribose, DD is di-deoxyribose nucleotide triphosphate. This is the abbreviation that we will be using from now. So, back here B2, here as usual you have the phosphate, and here you have 2 hydrogen. Catalyst, you use the polymerase, it is the same reaction as it goes, and there will be this formation of the phosphodiester bond here, one base would be attached. What next? This does not have a free hydroxyl group at the 3' end. Since this does not have a hydroxyl group, it cannot do the further process. So, it will stop the replication. In your solution, once this molecule is picked up by the polymerase and this reaction happens, after that your replication would be stopped, or extension, whether you call extension or replication, will stop. It cannot move any further. That is where the chemistry is involved, and that is where the beauty of it. So, Sanger's method is known as di-deoxy method. It also is known as a chain termination method. Chain termination, because as you have seen, it stops the replication in between. So, the complementary sequence or complementary chain, DNA chain would be terminated, would be stopped. So, that is what the entire idea is. Now, if I come back here, what Sanger has done, so here you have, I am not writing the all of this here. Additionally, along with all the 4 DNTPs, Sanger has used a little bit quantity of this DD thing for this particular vial, and here comes the differentiation. DD ATP was added in this case. So, vial 1 had all the 4 DNTPs plus the DD ATP, and now I call this vial, instead of 1 I call this vial A vial, because it has ATP added, DD ATP that was added. Similarly, vial 2 along with everything that it had, another DD, let us say CTP was added. So, this is A vial, I call this now a C vial. In this case, DD maybe TTP was added. So, this is a T vial, because it will be easier to figure out, and for this plus DD GTP, we call it G vial. So, here comes the difference between the vials, each vial is now different. And now, let us see how the reactions proceed, what good this is going to do, or this addition of the new DD NTPs are going to do. So, there was another trick, that is the Sanger has labeled this phosphate with a 32 radioactive element of the DGNTP. So, nowadays we do not use radioactive labeling, most often we use the fluorescence labeling, and that is done on the nucleobase, not here. So, anyway, be it the radioactive label, be it the fluorescence label, the labeling was done on the DDNTP, all of them. What is the use? The labeling is done to locate a thing, to track where the molecule is going. For example, if you keep your GPS on in your mobile, it is to know where exactly you are. People can track you, or you can know your location, where exactly you are. So, it basically to track somebody or to track something that we do labeling. Here also, so the radioactive labeling was done on P32 to track the molecule. So, all of these now has a star with P32. And here P32. And now let us take one vial at a time and let us see what are the reactions that are happening. So, this is basically here this. So, you have the DNA, you have the primer, you have added initially all 4 DNTPs ATP, TTP, CTP, GTP, you have added DNA polymerase 1, then it was divided into 4 individual vials, and in each vial a separate DDNTP was added. This is the structure of the DDNTP. It is a nucleobase, 2' deoxy, 3' also deoxy. So, it cannot go further extension for the when the synthesis of the complementary strand would be happening. So, usually done by controlled termination or interruption of the replication. That is the whole idea behind the Sanger's method. So, let us take a target DNA 5' and maybe I am taking an arbitrary sequence ATGC, ATGC, ATGC 3'. For the sake of simplicity of, for me it would be easier if I write it on the other way around. So, my target DNA single stranded DNA I am writing now from the 3' to 5' end, it would be easy for me. C, G, T, A, C, G, T, A, C, G, T, A. So, this is the target DNA. Let us consider vial A, where you had added the DD ATP and that had a radioactive label. So, what will happen? Let us say there was something else here, the other part of the DNA that we are not considering, and here the DNA primer was bound, because you had to start with a DNA primer. This one was the DNA primer. So, we will not consider that part now. We will see what happens after this, after the primer, after you start your reaction basically. So, this is your 5'. Here it is basically starting with the 5'. Now, what will happen? You have all the 4 DNTPs, you have polymerase and you have the primer ready. So, it will start the extension. So, it will have the first G that will be picked up against C, then it will be C here, then there has to be one A here. Now, here comes the trick, or here comes the difference of reactivities, that you have added the normal D ATPs there in the vial A, that was there. So, if it takes the normal DATP that has a 3 prime hydroxyl group free and it can move on. So, if it takes up, if the polymerase picks up the DATP, it will move on forward direction, no problem. But instead of the normal DATP, if it picks up the di-DATP, because you have also added this compound into your mixture. So, there will be competition between the normal ATP and the di-DATP, and it may happen, that is a high probability of course, that certain molecules or in certain molecules the di-DATP will be picked up, and once this is picked up you have a 3 prime hydrogen here. So, your chain is terminated, there will be no further extension, and that is where the labeling is also coming. So, since you have the labeling, it will amplify the signal and you can track where this molecule is now. Okay. So, what Sanger has done, in each of these vials that you have added the DD-NTPs, the quantity of the DD-ATPs or DD-CTPs, DD-NTPs were small compared to the normal DATPs or the normal DNTPs. The idea is you allow the extension to happen, but at the same time it can stop the certain number of molecules or fraction of the reactions can be stopped due to presence of these molecules. So, if you have for example 100 copies of DNA or 200 copies of DNA at the beginning, then what will happen, maybe 10 copies of DNA will be stopped here and rest 90 or rest 190 will move along. That is where you get the more fragments. So, in this case, the case number 1. So, first case is that your reaction may stop here. Case number 2, I am not writing the primer again anymore, I will move directly here. G, C, that it takes up the normal DATP, so it will move along. Here is a T, here would be G, here would be C, here would be A. Again the same issue, that this A may be the normal DATP, so it will move on, and the probability is also there that it may be the di-DATP. So, in this case it will stop here. So, you will get the 2 fragments and they are radioactive labeled. Case 3, 5 prime G, C, A, T, G, C, A, T, G, C, again A has to come and it may stop right here, and case 4 that it does not stop at all, A, T, C, G, C, A, T, I think it should be G, C, G, C, A, T, G, C, A, T, G, C, A, T. So, I am sorry, this should be G, this should be C, G, C, A, T and G, C, A, T, G, C, A, T, G, C, A, T, G, C, A, and then finally T, hydroxyl. But this molecule does not have any label here. So, you will not see this and it will not interfere into your data because there is no tracking here. So, it does not give you any signal even if it is present. So, ideally with radioactive labels you get 3 different fragments, one this, one this, one this. Now, we will use this just in a bit time. So, ATGC, ATCGTA, right, 3 prime, CGTA, CGA, T A C G T A. So, now come to the second vial. Second vial was your CTP, I guess, here CTP. So, we call it C vial. C vial, you have DD CTP with radioactive level. Start with the 5 prime. I am not showing that this is a primer, this is the other part of the DNA, this is the DNA primer. So, it has to take a G, which will be a normal G. Now, it has to take a C. So, again this C can be the DCTP, or if it can be the DG-CTP. If it is DD-CTP, then this will stop. It has a hydrogen at the 5 prime. So, further elongation is stopped, and you have the radioactivity level. So, this is 1, it is case 1. What can be 2? 5 prime GC, that it goes on AT GC again here. So, wherever there is a probability of picking up a C, here maybe a DD-CTP can be picked up and chain will be stopped. 5 prime GC AT GC AT GC, stopped. And then the fourth one is the full length, I guess, T G C T G C A T, and a hydroxyl group here, but again this guy you cannot see because it does not have a label. So, 3 fragments basically also from this vial, CGTA, 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 CGTA. So, vial, what was the original one? I am following this. So, TTP, T vial, and then will come the G. Vial T. So, we call it T vial, where you have added DD TTP labeled. So, case 1 5, this is GC A T. So, wherever there is a probability of taking up a T, it can be either the normal T or it can be the labeled di-deoxy T. Once it is there, it is hydrogen, the chain is terminated. Case number 2, GC A T, GC A T star. Five prime G C A T G C A T G C A T, the last one, and it would be H, and of course, that is the full length. So, again 3 cases. So, I think here itself we can do the vial G, where we had added DD GTP. So, here first case would be 5 prime G, the very first base, and that would be stopped, no further. So, please remember that there are other sequence before this that I have not considered, and I am assuming that the DNA primer that we have used would be complementary to that sequence for all cases. G C A T, here there has to be another G, stopped, 5 prime G C A T G C A T, here there has to be G, and the full length I think 5 prime G C A T G C A T G C A T OH, and again this is not labeled. So, now you can see so many fragments you have. Now, here comes the chemistry. Since you have fragments, now you can study them. The whole long DNA is hard to study at a time. So, now if you have the smaller sequences, you can study them well. Another thing, did you notice that all of these sequences have different length of the DNA? For example, this is 1 nucleobase long, this is 1, 2, 3, 4, 5 nucleobase long. If you look at all the fragments that we have done, all of them are of different length of DNA. So, since DNA has negatively charged phosphates, so overall charge of the DNA is negative. So, number of phosphate means the number of negative charges. So, as the length of the DNA will increase, the number of charge will also increase, negative charge will also increase. And of course, by the charge density, now you can separate the molecules. If we call the fragments as individual molecules, now all these molecules now have different charges, and therefore, we can easily separate them. It is just like doing your organic chemistry. If you remember your basic organic chemistry, that we do thin layer chromatography, TLC, right, to see the mobility of compounds. And if you have a plate, for example, a silica plate or an aluminum plate, and if you have organic molecules or a sample which has mixture of compounds, and you want to know how many components there are, what is their polarity property. So, if this is compound A and if this is compound B, and if they have difference of polarity, then if you put a drop of this molecule here, if you put a drop of this molecule there and allow them to move, then they will move along the plate, and you can see depending upon their polarity, their degrees of movement or the speed or the velocity of their movement would be different. So, over certain time, for example, this compound will move until here. On the other hand, this compound will move in a more length, will move faster, and that is because they have different polarity. So, more polar molecule, in this case the polar molecule moves slower, non-polar molecule moves faster. It is also a kind of the charge difference. Same for DNA, that the more number of negative charge they have, in this case it is a little bit other way around, more number of negative charges it has, it will move faster, and that is called it is also a plate, it is also a kind of a chromatography, but this is called electrophoresis, gel electrophoresis. So, this is called gel electrophoresis. It is also known as autoradiogram. So, it is a electrophoresis technique, and any electrophoretic technique means they will separate molecule in terms of charge, or in principle m by z value, charge density basically. So, for DNA, the longer the DNA is, movement would be higher. So, now if you have the mixture from the A vial, from C vial, from T vial and from the G vial, if you put that certain quantity here, 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 here from the individual 4 vials, and you allow them to move, then according to the length they will move, and you have the tracker, the P32 labeled, or the fluorescence level, where you can see how far they have moved. So, that is where is done for the analysis. And let us say we divide the y axis in terms of the length of the DNA. This is 1 base long, this is 2, this is 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12 here. So, you have scaled your y axis to see the number of. Now, look at the A vial and let us see which lengths you get the spots. It is very simple. A vial you have, this is 3. So, 3 bases, I am writing 3B. So, this is for 3, this is 1, 2, 3, 4, 5, 6, 7 base. 3, 7, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This is your 11. So, 3, 7 and 11 you should see the spots. 3 here, 7 here and 11 here. So, you should see since you have 3 fragments, they should have different movement and it should come in these positions. And of course, the full length does not have the tracker. So, you cannot see DNA is invisible, the quantity is very low, or also DNA does not, normal DNA does not have any color. So, you cannot see that, all you will see where there are the label. So, 3, 7 and 11. Now, C vial, what you have? 2, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 2, 6, 10, 2, 6 and 10, 3 fragments. T vial you have 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So, that is the full length 4, 8 and 12, 4, 8 and the last one 12. Quickly for the G, 1, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7, 8, 9, 1, 5, 9, 1, 5, 9. This is your data, this is your result. Now, if you see here, every length is here, you have spot for every length. What you have to simply do is just read the gel and put the corresponding base here. So, we have started, basically we are actually synthesizing the complementary strand from the 5 prime end, right. So, this is your 5 prime end. Where is your first base? Here, G. So, this has to be a G. Second base is C. So, there is a C here. Third base is A. So, A. Fourth is your T. Fifth is your G. 6 here C. 7 A, 8 T, 9 is G, 10 C, 11 A and 12 is your T. This is the 3 prime. Now, this is so simple to analyze the data. By the way, this is your sequence of the complementary strand that you have been synthesizing. So, you can figure out now 5 prime GCAT, GCAT, GCAT. So, your original strand, so 5 prime GCAT, GCAT, GCAT. So, this was the complementary strand. So, your original strand or your target, the sequence of your target DNA would be of course CGTA, 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 CGTA. Now, let us match what was your target CGTA, CGTA, CGTA. So, just by reading your data, just by reading the gel, you can find out the sequence of your desired DNA. This is so simple analysis. So, if you think of small organic molecules and if you want to characterize, if you want to know the structure of the compound, what we do, you have to do maybe elemental analysis, you have to do NMR, you have to do FTIR and so on. And all these techniques require a lot of knowledge or require hard work to analyze the data itself. Then you you figure out the structures. So, this is something which is so simple to interpret. The data does not need much hard work or much knowledge to analyze. It is so simple. You would just see the gel and put up your DNA strands. This is what the beauty of the Sanger's method. Thank you.